Hi, this is a lecture for my microbes and their human host class. It is bacterial anatomy and physiology, and this is part 1. Today we're going to be talking about bacteria. So a little on the words. The bacteria, like at the very top of the slide, that is a plural word. So bacteria is a plural word. We're referring to many. Bacterium is the singular. So we don't want to confuse those two. Sometimes students will write or say bacterias and with an apostrophe S, and that is not correct because bacteria, that term is already plural. So try to use bacteria when you're referring to many, bacterium when you're referring to just one. Okay, a little background on bacteria. Bacteria are single-celled prokaryotes. Remember, we talked about the prokaryotic cell and how they're different from eukaryotic cells. Bacteria have specific shapes, so they're all a little bit different. You'll see them as cylindrical or spherical or even spiral shaped, and we'll talk about those in a couple minutes. They have a rigid cell wall that contains a molecule that is unique to bacteria. So we only find it in bacteria, and that's called peptidoglycan. You'll learn more about that also. Some bacteria will move around, and how they do that is with an appendage called flagella. They multiply, they have asexual reproduction, and they will divide by binary fission. Now, bacteria, where do they get their energy to stay alive? And they get it from a variety of sources. We'll learn about that when we talk about the metabolism, and even some are photosynthetic. Now, bacteria do come in a variety of shapes, but cells of a particular species are only of one shape. So, for example, Staphylococcus aureus. All the cells of Staphylococcus aureus are going to be of that one shape. So each species has their own shape. All right, so on this slide, you're able to see some of those shapes. You see some pictomicrographs, and then we also have some other types of shapes. Now, the one that is spherical, we call that a coccus. Coccus is a singular term. It's coccus for singular, it's cocci for plural. Then we have one that is a bacillus. So a bacillus or bacilli for plural, that one is a rod, and I tend to call them rods, but they are rod shapes. They kind of look like hot dogs. Some are a lot shorter though. There is one, a type of, there was a shape that is in between a coccus and a bacillus, and we call that a coccobacillus. So it's not quite spherical, but it's not really long like most of the bacilli. Okay, so that's coccobacillus. Then we have ones that are like Vibrio, and Vibrio reminds me of a comma. So those kind of look like commas to me. There are Spirillum. So Spirillum is curved rod, and it's long enough to form these little spirals. Then we have one that's called a spirochete. So a spirochetes are usually long, they have flexible cell wall, and those are the spiral ones. So you see a picture of a green spirochete there, and above that picture, at the very top of your slide, you'll see those spirochetes. So those are kind of cool. So these are the various shapes that bacteria come in, and I should point out these are the most common shapes that we find bacteria in. There are actually more shapes than this, and they're put into, kind of the others are put into a group called pleomorphic. So there are little oddballs that look different than the norm. We call those pleomorphic. So there are some other shapes, but these are the most common that we find. Now we're going to start really taking a look at the components that make up the bacterial cell. We're going to look at the layers of the cell, and we're basically going to work our way from the inside out. And if you're looking at the picture on the slide of the bacterial cell that's been opened up, we are going to start with that layer that looks like it's a brown color, okay? Now, that layer is called the cytoplasmic membrane. An older term for that is the cell envelope. Now, this cytoplasmic membrane, it is made up of a phospholipid bilayer, and you are very familiar with phospholipid bilayer because your plasma membrane is made up of a phospholipid bilayer with embedded proteins. Now, take a look at the other picture that I have up on the screen, and it looks very similar to your plasma membrane. So it has the same components, and therefore it's going to have the same characteristics. So for example, selectively permeable. So it is selectively permeable. Do you remember what that means? That means depending on the molecule, certain molecules are allowed to enter and exit through that membrane because of the properties. Now, how do these molecules get in and out? They get in and out via diffusion, okay? Remember, diffusion is a passive process. Osmosis, do we remember what osmosis is? Yeah, the movement of water, okay? That's also a passive process. And then we have active transport. So active transport can go on through the cytoplasmic membrane. Now, up until this point, it sounds almost exactly like your plasma membrane, the cytoplasmic membrane of bacteria. However, there is one big difference. There is a process that goes on in the bacterial cytoplasmic membrane that does not go on in your plasma membrane. So bacteria actually have an electron transport chain that occurs in their cytoplasmic membrane. So if you remember, the electron transport chain in our cells occur in the mitochondria. But in bacteria, it occurs in their cytoplasmic membrane. All right, on to another layer. Remember, we're working our cells from the inside out. Now take a look at the picture on your screen. We already talked about the cytoplasmic membrane, which is represented in brown. Now we're moving to the one that represented in blue. So that is called the cell wall. Now bacteria have cell walls, plants have cell walls. Oh, we don't have cell walls. So this might be a new concept for you. So a cell wall. The cell wall is a strong but somewhat rigid structure that really prevents that cell from bursting, okay? Now, what's important about this cell wall is that it does contain that unique molecule that we only find in bacteria called peptidoglycan. So it contains peptidoglycan. Now, a guy by the name of Hans Christian Graham developed a staining process, and as a result of that, we were able to see two major types of bacteria, and that staining process separated them based on their cell walls. So we call that the Graham stain. In lab, you guys are going to learn how to do that Graham stain. So in Graham stains, it distinguishes between two main types of bacteria based on their cell wall. You can have Gram positive or Gram negative. So if you're looking at the pictures on the screen, Gram negative turns out kind of a pinkish color. Gram-positive is always a purple color. All right, so we are going to now talk about gram-negatives, gram-positives, then gram-negatives, and how they're different in their cell walls. All right, so now we're going to talk about the components and characteristics of gram-positive cell wall. So these are gram-positives. Now, one characteristic is that they have a thick peptidoglycan layer. Now, hopefully you can see my little laser pointer, and we're looking at the picture on the screen with looking specifically at the one on the left. So where you see these blue and purple balls, okay, these circles, that is representing the peptidoglycan. Okay, this is representing the peptidoglycan in that cell wall. Just for reference, down here at the very bottom is representing that cytoplasmic membrane. Okay, you should recognize that with the phospholipid bilayer. So this is the peptidoglycan layer. Now these red strings, what looks like red strings through them, that is another component called teichoic acid. So the teichoic acid will actually stick out from the top of the peptidoglycan layer on the external surface of that cell. The teichoic acid is thought to be important in cell wall construction and also cell division. Now, in between this thick peptidoglycan layer and the cytoplasmic membrane, we find a gel-like substance called periplasm. Okay, so it's in between, there's a periplasm layer in between the cytoplasmic membrane and that peptidoglycan layer and teichoic acid. Both of that together makes up the gram-positive cell wall. Now, some components and characteristics of gram-negative cell walls. So we're really going to compare the two. So first of all, that picture looks way different. You probably recognize some of those components, hopefully you do, of some that we just talked about in gram-positives. But gram-negatives look way different. So first of all, for number one, let's look at the peptidoglycan layer. So right in the middle, you should recognize the blue and purple circles that's representing the peptidoglycan layer. Okay, it's not as thick as it is in gram-positives. So gram-negatives have a thin peptidoglycan layer. And they have something that gram-positives do not have. They have an outer membrane, okay? So they have an outer membrane made up of a phospholipid bilayer, okay, and some other things going on. So this outer membrane is a unique lipid bilayer. It is physically joined to the peptidoglycan layer, and you can see that represented in these purple, kind of purple spikes, we'll say. Because of this outer membrane that gram-negatives have, that outer membrane can actually block some molecules from getting to the inside of that cell, which can be advantageous for that cell. So for example, there are some antibiotics that cannot make it through that outer wall, and as a result can't get to the inside of that bacterial cell, so it has no effect on that bacteria whatsoever. All right, so next we're going to learn a little bit more about this gram-negative cell wall. So on to the next slide. All right, we know this outer membrane that gram-negatives have. It's a phospholipid bilayer, you recognize that. But notice all of these little red wiggly things that are sticking out from on the outside surface. One is circled, and we're looking at this picture at the top right. Now, that shows us that that is made up of a few different molecules, a couple different molecules. That in itself is called lipopolysaccharide. So this entire molecule here is called lipopolysaccharide. And a lot of times in microbiology, they like to abbreviate. So it's known as LPS. So LPS stands for lipopolysaccharide. Now, a few things about lipopolysaccharide. Our bodies will react to this lipopolysaccharide, and it will be it's referred to as endotoxin. So when our bodies recognize it, it's recognized as a toxin. Now there are two major parts to lipopolysaccharide. So one part is called lipid A. And on the picture, the lipid A, I don't know if you can actually, if you can read it, but the lipid A is this portion, the bottom portion of that molecule. Lipid A is the part that's recognized as a toxin, okay? The other part of that molecule is an O antigen. And the O antigen, we use that part to identify the strain, the type or the strain of bacteria. So gram-negatives do have a thin peptidoglycan layer. They have the outer membrane. They have some pretty significant molecules associated with that outer membrane, lipopolysaccharide. And the last thing I want to point out to you about the gram-negative is the periplasm. So it has periplasm surrounding that peptidoglycan layer. So we're going to find periplasm between the cytoplasmic membrane and that outer membrane in gram-negatives. All right, so it's pretty interesting stuff when we're talking about the cell walls of bacteria. So gram-positive cell walls, gram-negative cell walls, and as it turns out, some bacteria don't even have cell walls. Sometimes they have a cell wall and then they lose that cell wall. So for example, Mycoplasma, there's a species known as Mycoplasma pneumoniae. It causes a mild form of pneumonia, what's called a walking pneumonia, and it will actually lose its cell walls. It doesn't have a cell wall, and it evades our immune system that way. Okay, some other possible layers that could be associated with bacteria. And those are the capsule and the slime layer. We're first going to talk about those. So capsule and slime layer. So first, number one, capsule. The picture I have up on the screen, look at the one that has the orangish or brown around it. So that's representing a capsule. And it's kind of like a gel-like layer. Okay, and then, or we can have a slime layer. A slime layer is more irregular, it's more diffuse around that cell. And really we're going to, for our purposes, for me, I'm going to lump those two together. So capsules and slime layers. So these layers are outside of the cell wall, okay? So they're more exterior to the cell wall. And overall, it helps to protect the cells and also allows for attachment, allowing the cell to attach to other surfaces. What makes them up? They're either made up of glycocalyx or polypeptides. Now, these components, these layers, will allow bacteria to grow as a biofilm. You're going to learn much more about biofilms later, but biofilms will, it's kind of a community of bacteria living together. All right, so, and also one more thing, some capsules allow bacteria to evade our host immune system. So in having that capsule, kind of, it's like a cloak. It's a cloak where our immune cells don't even recognize it as being bacteria. All right, so other possible structures. These are external structures. One is a flagella. So if you remember early on in this lecture, I talked about bacteria, how they use flagella in order to move around. So some bacteria have flagella. So some species will have flagella, others won't. If it does have flagella, then it's able to move around or be motile. So motility means the ability to move. So motile means move. Now, different types of flagella. So we have a trichus. The word trichus means flagella. You want to think of that as meaning flagella. So with the A means without. Without flagella, those species without flagella just do not move around. All right, so flagellar arrangements. There are species that just have one flagellum on one side, and they're called monotrichous. Mono means single, so single flagella. Okay, so other types of flagellar arrangements, you have lophotrichous, so many on one side, on one end. You could have amphitrichous. Amphitrichous, you have flagella on both ends. And then peritrichous. Peri means surrounding. So the flagella surround the cell. So I do want you to know the arrangements of flagella. So if I asked you which flagellar arrangement is when the bacteria have many flagella on one end, what would be the answer? Lophotrichous, that's right. So I may ask you to describe a bacteria that has the arrangement of peritrichous. That's right, flagella that surrounds the cell. All right, so I have a link to a very short YouTube video where they filmed bacteria moving with flagella. There are some microscopes, some types of microscopes that can be used to see live bacteria moving around. And sometimes you can see the flagella. So this link on the screen will allow you to see that filmed from a microscope. It's pretty cool. I'll also put that link down in the comments of this video as well. So click on it, take a look at it, and then move on to your next slide. All right, a couple other external structures of bacteria. All right, another one is pili. So pili is a plural word. It's pilus for singular. What these are used for is mainly for connection to other cells, used for DNA transfer. Bacteria produce asexually, but we will talk about the times when DNA transfer can occur in bacteria, and they do use a conjugation pilus. Now, the picture I have up on the screen, you'll notice there are two cells there, and there is a conjugation pilus. So these basically are protein tubes that are made by one cell and extend to another. So for the most part, the pili are used for DNA transfer, connection to other cells, but more secondarily, they could be, on certain instances, used for motility. Okay, a third one is called fimbriae. So fimbriae are used for a surface attachment. So surface attachment for the bacteria. Now it's a little more faint on our picture, but you can see that fimbriae surrounding the bacterial cell. So fimbriae, think of they kind of resemble long, thin hairs extending from the cell, but it's not hair, and it's used to help attach them to surfaces. All right, so those are the external structures, possible external structures, and this concludes our part one of Bacteria, talking about bacteria anatomy and physiology.